Hello everyone, today we'll discuss about carbon dioxide transport. As we all know, due to metabolism, carbon dioxide is produced in our body cells. This carbon dioxide is transported from this tissue cells to the lung, and this transport is facilitated by blood circulation and diffusion of the gas along the pressure gradient. Now we discuss about the pressure gradient. You can see here. Intracellular partial pressure of carbon dioxide is 46 mm of mercury. Interstitial fluid partial pressure of carbon dioxide is 45 mm of mercury. In the arterial blood, partial pressure is 40 mm of mercury. In venous blood, it is 45 mm of mercury. And you can see here. And in the alveolar air, partial pressure of carbon dioxide, that is again 40 mm of mercury. Therefore, carbon dioxide is transported from cell to the interstitial fluid, from interstitial fluid to the arterial blood, and from venous blood to the alveoli. Now, pressure gradient, carbon dioxide is transported from the cell into the interstitial fluid along a pressure gradient of only 1 mm of mercury. You can see here this is 46 inside the cell and 45 mm of mercury in the interstitial space. From the interstitial fluid to the venous blood, carbon dioxide diffuses at a pressure gradient of 5 mm of mercury. You can see here interstitial fluid partial pressure of carbon dioxide is 45 mm of mercury, and in the arterial blood it is 40 mm of mercury. And from venous blood to the pulmonary alveoli, it diffuses again along the partial pressure of 5 mm of mercury. You can see in the venous blood partial pressure of carbon dioxide is 45 mm of mercury, in the alveoli it is 40 mm of mercury. Now, diffusion coefficient for the carbon dioxide is 20 times more than the diffusion coefficient for the oxygen, and this is the reason only this much gradient of the carbon dioxide is sufficient for its transport. Now, transport of carbon dioxide. Carbon dioxide is transported in 3 steps. Number 1, that is diffusion of carbon dioxide in the blood. First of all, carbon dioxide enters inside the blood from the cell. Second, that is transport of the carbon dioxide in the blood. And third, this carbon dioxide from blood, it is released in the lungs. We will start with the first step. Diffusion of carbon dioxide in the blood. As we all know that cell metabolism produces carbon dioxide. You can see here in the tissue cell, because of metabolism, carbon dioxide is produced. And therefore, partial pressure of the carbon dioxide is about 46 mm of mercury in the tissue cell. Okay? Now, in the interstitial fluid, partial pressure of carbon dioxide is 45 mm of mercury. So now here along this pressure gradient, carbon dioxide enters into the interstitial fluid, and from interstitial fluid now it enters into the blood, as we have already discussed. Okay? So in the venous blood, partial pressure of carbon dioxide, now it becomes 45 mm of mercury. You can see this venous blood is now been transported. Another important thing, at partial pressure of carbon dioxide 45 mm of mercury, carbon dioxide content of the venous blood, here pay attention, this is partial pressure. Partial pressure is 45 mm of mercury, and at this partial pressure, carbon dioxide content, that becomes 52 ml. Okay? And the blood, which is arterial, it has partial pressure of carbon dioxide 40 mm of mercury, and in arterial blood, carbon dioxide content, that is 48 ml. So how much is the difference? Difference is 4 ml. Now this 4 ml of the carbon dioxide is excreted per 100 ml of blood. So you can see here how much carbon dioxide is transported. As we have discussed, in the venous blood, carbon dioxide partial pressure is 45 millimeter of mercury, but at that partial pressure carbon dioxide is present about 52 ml per 100 ml of blood, whereas in the arterial blood, when partial pressure of carbon dioxide is 40 millimeter of mercury, carbon dioxide content, that is 48 ml per 100 ml of blood. So the difference, that is 4 ml of carbon dioxide per 100 ml of blood, that is being transported. Now we discuss about carbon dioxide transport. Carbon dioxide is transported in three forms. One, that is dissolved form, which is 7% of the total transport. As we have discussed, how much is the total transport? 4 ml per 100 ml of blood. So from 4 ml, about 7% is in the dissolved form. So you can see 7% of the 4 ml, that is equal to 0.3 ml per 100 ml. Second form of transport, that is bicarbonate form. This is 70% of the total transport. 70%, that is 70% of 4 ml. It is equal to 2.8 ml per 100 ml of blood. Bicarbonate form is being transported. Third, that is carbamino form. That is carbon dioxide combines with the amino acid, and here this is 23%. 23% of 4 ml, that is equal to 0.8 ml per 100 ml of blood. Now you can again see here, this is the same thing, 7% in the dissolved form, 70% bicarbonate form, and 23% in the carbamino form. Carbon dioxide is transported. Now starting with dissolved form. As we all know, venous blood partial pressure of carbon dioxide, that is 45 millimeter of mercury, and in venous blood about 2.7 ml of carbon dioxide is present per 100 ml of blood, and in arterial blood partial pressure of carbon dioxide is 40 mmHg and dissolved state, that is 2.4 ml per 100 ml. So the difference, that is 0.3 ml per 100 ml, that is transported in the dissolved form. Now from this 0.3 ml per 100 ml of carbon dioxide which is transported in the dissolved form, about 0.2 ml per 100 ml of carbon dioxide, that is transported in the plasma, and only 0.1 ml per 100 ml of the carbon dioxide is transported through RBC. Although it is a very small amount, but this reaction is fast in RBCs. Why? That is because of presence of an enzyme carbonic anhydrase. So this is the first form of transport, dissolved form. Second form of transport, that is transport in the bicarbonate form, that is about 70%. 70% of carbon dioxide is transported in bicarbonate form. Mainly this reaction takes place inside the RBC because of, as we have discussed, presence of carbonic anhydrase enzyme. Now what happens inside the RBC? Carbon dioxide enters inside the RBC. You can see here this carbon dioxide combines with H2O and it forms carbonic acid. Now this carbonic acid, that is again further converted into H plus and HCO3 minus. Now this H plus ions, they bind with the hemoglobin and it forms HHB, deoxygenated hemoglobin. Now what happens to this HCO3 minus? This HCO3 minus, they diffuse out, and this HCO3 minus transport, that is associated with the transport of chloride ions, and this is known as chloride shift. This reaction can also take place in the plasma, but due to absence of carbonic anhydrase enzyme, this reaction is slow. So carbonic anhydrase enzyme which is present in the RBC, it catalyzes the reaction and it accelerates this reaction, and the rate of the reaction is increased about 5000 folds inside the RBC. It is proven by administrating carbonic anhydrase inhibitor like acetazolamide. If we are administrating the drug, it blocks the carbonic anhydrase, and because of blockage of carbonic anhydrase, CO2 transport that is reduced. Okay. Now, inside the RBC, as we have discussed, this reaction has taken place. This H2CO3 is converted into H plus and HCO3 minus. Now, this H plus ions, as we have discussed, they are buffered by the hemoglobin, and that prevents the reaction to occur in the opposite direction. And as a result of this reaction, hemoglobin is in the reduced form. But bicarbonate formation is rapid, as we have discussed, because of the presence of the carbonic anhydrase enzyme. Now, chloride shift, which is also known as hamburger phenomena. Why? Because it was invented by Hamburger in 1918. As the blood passes through capillaries, as we have discussed, when carbon dioxide enters and it is converted into carbonic acid, and which is further converted into H plus and HCO3 minus. So, when blood passes through the capillaries, HCO3 minus ion concentration increases inside the RBCs, and this HCO3 minus ions, they enter from RBC into the plasma. Why? Because inside of the cell becomes negative, and to decrease the negativity, this negative ions, they move out. When bicarbonate ions, they move out, there is entry of one chloride ion inside, and that is known as chloride shift. Now, as a result of chloride shift, number of ions inside the RBC increases, and therefore this RBCs, they take up more water. That is known as osmotic absorption of fluid inside the RBC, and therefore osmotic pressure of RBC increases. And therefore you can say venous blood RBCs, they contain large amount of fluid as compared to arterial blood RBC, and it is the reason why hematocrit of the venous RBC is about 3% greater than the arterial RBC. Also, venous blood RBCs, they are more fragile as compared to arterial RBC. This we have already discussed. Chloride shift, bicarbonate ions and chloride ions, they move in opposite direction. Now, third form of transport, that is transport of carbon dioxide in carbamino form, that is 23% of total carbon dioxide or 0.8 ml per 100 ml of blood. Now, inside the plasma, carbamino compounds, they are formed, and these compounds are formed by combination of carbon dioxide with amino group of the plasma protein, and it forms carbamino compound. This reaction is less significant. Why? Because quantity of this proteins, that is less. It is about 1/4 than that of the hemoglobin, and therefore this reaction is less significant. Now, inside the RBC, this carbamino hemoglobin is formed, and it is formed when carbon dioxide combines with the amino group of the hemoglobin. There is formation of carbamino hemoglobin. This carbamino hemoglobin formation is also fast and reversible reaction, and about 0.6 ml per 100 ml of carbon dioxide is transported in this carbamino compound. This reaction is also a rapid as well as loose reaction, which means carbon dioxide can be easily released in the alveoli when partial pressure of carbon dioxide is less than the tissue capillaries. Now, carbon dioxide dissociation curve. It shows the relationship between partial pressure of carbon dioxide here and carbon dioxide content. Now, this relationship, that is linear. In arterial blood, when partial pressure of carbon dioxide, that is about 40 mm of mercury, at that time carbon dioxide content, that is 48 ml, here it is. And in venous blood, when partial pressure of carbon dioxide, that increases and reaches up to 45 mm of mercury, here you can see this one. At that time carbon dioxide content becomes 52 ml per 100 ml of blood. Now, factors affecting carbon dioxide dissociation curve. First and foremost, that is oxygen. Deoxygenated hemoglobin that can load more carbon dioxide than oxygenated hemoglobin. You can see here, this is the curve for arterial blood, this one. It is shifted towards right side. Whereas for the venous blood, the curve is shifted towards left side. Now we'll discuss. So deoxygenated hemoglobin has more carbon dioxide than oxyhemoglobin, and this effect is known as Halden's effect. In simple words, we can say that when oxygen binds with the hemoglobin, it dissociates carbon dioxide from the hemoglobin, and this is seen at the lung level, which is known as Halden's effect. Here you can see in the arterial blood, the curve is shifted towards right side, means at the same partial pressure of carbon dioxide, CO2 content that is less. You can see here, suppose at 40 mmHg partial pressure of carbon dioxide, in the arterial blood CO2 content is, you can see here, it is about 50 ml per 100 ml. But at the same partial pressure of carbon dioxide, if the blood is venous blood, in the venous blood, because partial pressure of carbon dioxide is the same but partial pressure of oxygen is less. You can see here in the venous blood, partial pressure of oxygen is only 40 mmHg. So what happens here? The curve is shifted towards the left side, means carbon dioxide content is more in the venous blood. The advantage of shift of carbon dioxide dissociation curve. Because of shift of carbon dioxide dissociation curve as well as Halden effect, double amount of carbon dioxide is released from the blood to the pulmonary alveoli, and double amount of carbon dioxide is released from the blood in the lungs. Second factor, that is increase in the 2,3-diphosphoglycerate. 2,3-diphosphoglycerate that is produced because of the metabolism. This 2,3-diphosphoglycerate compete with carbon dioxide for binding with the hemoglobin. 2,3-DPG binds at the same site where carbon dioxide binds, and therefore carbaminohemoglobin cannot be produced, and therefore carbon dioxide dissociation curve shifts to right, and therefore less amount of carbon dioxide is carried out when 2,3-DPG level increases. Another factor, that is temperature. When the temperature increases, carbon dioxide dissociation curve shifts to the left side, and large amount of carbon dioxide is taken at given partial pressure of carbon dioxide. So again you can see here, this is the carbon dioxide dissociation curve for the arterial blood, this is the curve for the venous blood, and the same curve for the venous blood when the person performs exercise. Next is release of carbon dioxide in the lung. As we all know, partial pressure of carbon dioxide in the venous blood, that is about 45 mm of mercury, and in the venous blood carbon dioxide content is 52 ml per 100 ml of blood, whereas in the alveolar air partial pressure of carbon dioxide is 40 mm of mercury and carbon dioxide content, that is 48 ml per 100 ml of blood. Therefore, difference of these two, 52 ml and 48 ml, that is equal to 4 ml of carbon dioxide is transported per 100 ml of blood. Now, how this carbon dioxide is released in the lungs? Release of carbon dioxide from the dissolved solution. Second, carbon dioxide is released from the carbonate into the plasma. Here, when oxygen enters inside the RBC and it binds with the hemoglobin, there is formation of oxyhemoglobin. This oxyhemoglobin, it is a strong acid, and this oxyhemoglobin which is formed, and according to Halden's effect, when oxygen binds with the hemoglobin in the lungs, it tends to release carbon dioxide. Because oxyhemoglobin has two effects. One, oxyhemoglobin has low affinity for carbon dioxide and therefore it releases carbon dioxide which diffuses into the plasma. And second thing is, oxyhemoglobin is a strong acid, stronger than the carbonic acid, and therefore strong acid releases H plus ions, and this H plus ions which are released, they combine with the HCO3 minus, you can see here. So H plus combines with the HCO3 minus and they form H2CO3 inside the RBC, and this H2CO3 is converted into H2O and CO2, and the CO2 is released. So you can see here there is reversal of chloride shift, means there is entry of bicarbonate ions inside the RBC, and when one negative ion enters inside the RBC, one negative ion exits out, and that is chloride ions, they exit out. Now here you can see this diagram shows reactions taking place at the tissue level as well as in the lungs. So at the tissue level you can see carbon dioxide is produced. Now this carbon dioxide, when enters inside the cell, this carbon dioxide combines with the H2O in presence of carbonic anhydrase, and H2CO3 is formed, which is further converted into H plus and HCO3 minus. This H plus combines with hemoglobin and it forms HHB, that is reduced hemoglobin, and this bicarbonate ions, they exit from RBC, and at that time there is chloride shift. All these reactions are reversed when the same blood reaches to the alveoli. Here you can see that when oxygen enters inside the cell, oxygen combines with hemoglobin, so hemoglobin becomes oxygenated. This oxygenated hemoglobin is a strong acid which releases H plus ions. Now this H plus ions combines with HCO3 minus ion. This HCO3 minus ions, they enter from plasma inside the RBC, and here you can see there is reversal of chloride shift. H plus combines with HCO3 minus ions to form H2CO3, and in presence of carbonic anhydrase, this is further converted into H2O and CO2, and this CO2 is excreted. So this is all about the transport of carbon dioxide. Thank you.